Alright, so in this video we're going to start off talking about the human life cycle with fertilization and mitosis. So these two steps right here. So fertilization occurs between a sperm and an egg, two haploid cells that have one copy of each chromosome, and they're going to come together to form a single-celled zygote. This zygote can then make up all the cells of the human body by going through mitosis, cell division. So we're going to do this with sock models, and I trust that you have this worksheet handy if possible. Yeah, so let's begin. So here in front of us we have a haploid cell. We have two autosomes and a sex chromosome. In this case we're going to use stereotypical coloration, quote unquote, and we're going to say that the blue autosomes come from the father and that the blue sex chromosomes are the Y chromosome. We're going to say that the pink sex chromosomes are the X chromosome, even though that biological males have both an X and a Y chromosome. So we have 3 chromosomes in front of us. We have 1 copy of each chromosome. So that makes this 1n equals 3. This is a haploid sperm cell because the autosomes are blue. So we have a blue sex chromosome. So this sperm happens to be carrying a Y. It could also be carrying an X because males have both an X and a Y. So what we're going to do is we're going to fertilize. We're going to fertilize an egg. Our egg is represented by these socks. Likewise, it has 2 autosomes and a sex chromosome. Eggs can only carry an X chromosome because females have 2 X chromosomes. So they can only donate an X chromosome to their offspring. Males can donate either an X or a Y. So by combining these, we're going to fertilize. So this is fertilization. So we're going to put these 2 together. We're going to put these 2 together. And we're going to put these 2 together. Right. So we now have 6 chromosomes in 3 pairs, 3 homologous pairs. Note that a pair is 2 items. It goes for socks, shoes, anything. A pair is 2 items. And a homologous set is a pair. So here we have 2 homologous chromosomes, each in 3 pairs. Alright, so this shows a diploid zygote. We have 2n equals 6 because we have 2 copies of each chromosome and 6 chromosomes total. So 2n equals 6. And so what's the biological sex of this zygote? It is male because we have an X and a Y chromosome. Note that that's quite different from gender. Alright, so how many chromosomes do we have? We have 6 chromosomes. What we're going to do now is we're going to go through the S phase of the cell cycle. So note that whenever we fertilize, we're going to start at G1. So now let's do the S phase, which S phase consists of replicating. So we're going to replicate every chromosome by pulling out the sock inside. So now we can attach them at the centromere, which is a little Velcro. Right, so we're going to do this for all of them. So DNA replication takes place during S phase of the cell cycle. This is a part of interphase before we get to the mitotic phase. Just like me pulling out all these socks, it takes a lot of energy of the cell to replicate all this DNA. Here's our X chromosome. And here is our Y chromosome. Notice how the Y chromosome is much smaller than the X chromosome. Alright. So now we have 6 chromosomes still in front of us, because remember, the number of chromosomes is dependent on the number of centromeres. So since these are attached, that's 1 centromere. So there's 6 chromosomes. However, we have 12 chromatids because each chromosome has 2 chromatids at this point. So now we're in G2. We're in G2 of the cell cycle. We've gone from G1 through S to replicate, and now we're in G2. So what we're going to do is we're going to begin mitosis. So we go through prophase, the nuclear envelope breaks down, we can't really model that here. And then we're going to go through prometaphase, that's where the nuclear envelope breaks down. Prophase is more the chromosomes condense. And then we're going to get to metaphase. And metaphase is whenever the chromosomes align at the metaphase plate. So I don't have a lot of room here, but I'm going to align them the best that I can. If you're doing this at home, I suggest you get a bigger table than me. And so we now still have 6 chromosomes with 12 chromatids at metaphase. However, we're going to begin anaphase, and anaphase is going to pull apart the chromosomes. So anaphase pulls apart the chromosomes, and chromatid disjunction is shown by pulling apart the Velcro centromere. And the second that this centromere is pulled apart, these are now their own chromosomes, because now they have their own centromere. They're no longer attached. So anaphase, that's chromatid disjunction. We're going to pull them apart. One fell off. That could be non-disjunction. I don't know. We're going to pull them all apart. Alright. So aside from the one chromosome that fell off the table into oblivion, we're going to say that we now have, so there's that cell, and there's this cell. So we could say that this is telophase, where they decondense and become all jumbled back up again, and the nuclear envelope reforms, and boom, there we go, telophase. So now we have 6 chromosomes in each cell, each with 6 chromatids, right? Because they're no longer replicated. So we have 1, 2, 3, 4, 5, 6. 6 chromosomes, only 6 chromatids. These will replicate again in the next S phase of the cell cycle. Alright, so comparing these cells to what we started with, we see that the zygote is exactly 50% maternal DNA and 50% paternal DNA. As we'll see in meiosis, the levels of grandparent DNA could be a little bit different. Following fertilization, the cell had twice as much DNA as each haploid sex cell did. And then notice how the zygote was identical to the 2 cells formed, and these 2 cells are identical to each other. They have the exact same chromosomes. So this is fertilization and mitosis. There will be another video on meiosis, which is a bit more complicated, but we will get through it.